Okay, dear students, in this video, we shall discuss about another type of asexual reproduction which is known as vegetative propagation. Okay, so you all might have heard about the term vegetative propagation. It is a type of asexual reproduction in which new plants are produced from the vegetative parts of the plants, which include the stem, root, leaf, etc. Okay, so here the new plants produced are identical to the parent plant since we are not using any sexual means. Here only asexual means are used and they are produced from the plant parts. And these plant parts are known as vegetative propagules. Okay, now basically we can say that the vegetative propagation is of two types. They are natural vegetative propagation and artificial vegetative propagation. So first let us move on to the natural vegetative propagation. In natural vegetative propagation, here we are using only the plant parts like the roots, stem, leaf, etc. And you can see that you have already studied that from roots, the plants which are propagated through roots include the sweet potato, carrot, then tapioca, etc. This we can see in the picture that is coming ahead. Then through stem there is potato, onion, ginger, etc. that is propagated. And through leaf there's a special name for the plant which is propagated through leaf, which is known as bryophyllum. Okay, so in all these cases you can see that here the root is acting as a medium through which new plants are produced, and the next one stem is acting as a medium to which new plants are produced, and leaf is also acting as a medium through which new plants are produced. And here you can see the pictures of the natural vegetative propagation. Okay, the next one is known as artificial vegetative propagation. This includes growing of plants by man-made methods or techniques, and the commonly used methods are usually of three types, which is cutting, layering, and the last one is known as grafting. Okay, so there are three types of artificial vegetative propagation which is made by man, which is known as cutting, layering, and grafting. So first let us move on to the first method, which is known as cutting. Okay, so you have to understand that it is a process by which a stem is given an oblique cut. Oblique cut means a sidewise cut, and then the cut stem is taken and it is kept separately under the moist soil. Okay, and we are planting the cut stem in the moist soil. After some days we can see that from that new plant arises. This is a common thing that we all use in our own homes just to plant a new plant. So whenever we feel like going into our neighbor's house, we see that a plant is there which is very beautiful, then we ask for a stem cutting. We take the stem cutting and we will plant it in our garden. Then after some days we can see beautiful plants will grow in our garden too. So that is a common thing we all use, that is the cutting. Okay, so you can see in the picture a plant is getting cut from one of the branches, and that branch is planted separately and a new plant is produced. Examples are rose, hibiscus, etc. Hibiscus is your shoe flower. Okay, so this is the process in cutting. Second process is known as layering. It is a process in which the stem, while still it is attached to the mother plant, is buried under the soil for a while till it produces new roots, and it is then detached from the mother plant and it is grown as a new separate plant. Okay, the examples are jasmine and lemon. You can see in the picture, that is, there is a mother plant and one of the long stem of the mother plant is tilted down, and that particular branch which is tilted down is buried inside the soil, and after some time you can see new plants are arising from the buried part. Okay, and then we will detach it and we will plant it as a new plant. Okay, so this is called as layering. The plant is growing in a layer wise, so it is called layering technique. Okay, then the third one is known as grafting. It is a method in which actually the cut stem of two different plants are joined together in such a way that the two stems will later grow as a single plant. Okay, so here we have to listen carefully is that the cut stem, if you are taking a particular plant, you're cutting the plant into two. Okay, The first part above the plant is forming the scion, and the cut stem below the plant that is forming the root will be known as the stalk. So you have the scion and the stalk. Okay, now here we are joining the two plants together and they are going to grow as a single plant, and the new plant will show the properties of both the plants. Okay, so once again I'll tell what is grafting. Here it is a method in which the cut stem of two different plants are joined together in such a way that the two stems will grow as a single plant. Okay, now the cut stem of the plant that forms the shoot is known as the scion, and the cut stem of the plant which is going to form the root is known as the stalk. Okay, the new plant will show both the properties of the scion and the stalk. For example, we are getting bicolored roses, hibiscus, and different varieties of mangoes are made by this grafting method. Okay, and then these two, the scion and the stalk, will be grafted together and they will be tied, wrapped properly, and it will be waxed so that they are airtight. Okay, and then you can see new parts will arise from that particular grafted portion, and then they will grow into a new plant. Okay, so these are the three important artificial methods of vegetative propagation, which includes cutting, layering, and grafting. Next, we shall move on to the advantages of vegetative propagation. See, this is a method which is allowing quicker and easy propagation of plants. So, a lot of plants can be propagated in a very small time period. Now, plants which are formed by this method will bear fruits and flowers much more earlier than those which are produced through seeds, because you know that seeds will take some time to we can say germinate, and then after that if they are not getting proper things they will not grow properly. So it is a very easy method to propagate plants within a short period of time. Third important advantage is that it makes the propagation of those plants very easier that have lost the capacity to produce seeds. Okay, you know that some plants cannot produce viable seeds. In that case, this vegetative propagation is a very good method to propagate those plants. And lastly, seedless plants can be propagated by this method. You might have taken grapes, etc., which are not having seeds. So how do we get those grapes? We are actually propagating it through vegetative propagation. So we don't have seeds in those grapes which are easy to consume. Okay, so these are the advantages of vegetative propagation. So with that, we have come to the end of the asexual method known as vegetative propagation. Next, we are moving on to the next method, the sixth method, which is known as spore formation. Okay, so let us see what is happening in spore formation. Now, in this method, the reproductive structures called spores will develop into a new organism. Okay, so here the reproductive structures are known as spores, which are going to develop into new organism. Now actually spores are very minute unicellular asexual reproductive bodies that are actually stored in specialized spore bearing organs known as sporangia. Okay, so you should understand that the spores are the reproductive units in those organisms, and these spores are kept very tightly intact in a body known as sporangium. Okay, now when the spores mature, the sporangium will burst open to release these spores. Okay, now after they are released, each spores will germinate and it will grow into a new organism. Okay, so that is how the spores are acting as reproductive structures. Example, you can see in the bread mold, which is called the rhizopus. It has thread-like structures called hyphae and a small knob-like structure called the sporangium, inside which the spores are they are kept intact. Okay, so when the favorable condition returns, the sporangium will break open and the spores are released. And these spores, when they fall on suitable substances, they will start to germinate and forms the new organism. Okay, so this is the technique of spore formation, which is considered as one of the asexual means of reproduction. And usually the fungus are the organisms that reproduce through spores. Okay, next we shall move to the last method of asexual reproduction, which is known as tissue culture. You might have already known about tissue culture because recently there are so many plants which are propagated through tissue culture technique. So let us see what is tissue culture. Actually, it is a method of cultivating plants wherein new plants are grown by removing a tissue or separating the cells from the growing tip of a plant. You have already studied that the growing tip of the plants consists of tissues which are known as meristematic tissues. So if these tissues are isolated and if they are kept in suitable medium, they can start producing mass of cells, and thus a plant can be grown. Okay, so the cells are then separated in an artificial medium containing the hormones called auxins and other nutrients, where they will divide rapidly to form a group of cells known as callus. So initially these meristematic cells will be growing into a group of cells which are known as callus. Okay, the callus will be then transferred to another medium containing plant hormones for growth and differentiation, as a result of which small plants are produced which are known as explants, and that are then placed in the soil so that they can grow into mature plants. Okay, so many plants are being propagated to tissue culture. So tissue culture, we can propagate a large number of plants within a very short period of time, and once we make sure that these plants have grown into the seedling state, then they can be safely transferred onto the field or other plant growing places. Okay, so examples are oil palms, date palms, tomato, banana, apple, etc. These are all produced by this tissue culture techniques. Okay, so you can see the diagram that is given here. You can see that a small tissue from the particular growing tip of the plant is cut, and then that is placed in an agar medium which is containing nutrients and growth hormone called auxin. Then after some days we can see small tiny plantlets will arise, which are then later are planted onto the field so they can grow in a very efficient way. So this is a technique of growing plants using cells or tissues. Now let us move on to the advantages of tissue culture. So the first important advantage is that it's a very fast plant growing technique, which is known as tissue culture technique. So we can have a large number of plants within a very short period of time. Second advantage is that by using this technique, disease resistant plants can be produced. This is because we are mainly taking the meristematic tissue, and since meristematic tissues are fastly dividing cells, most of the microbes cannot attack these meristematic cells. So when we are taking tissues from the meristematic region, we can get disease resistant plants. Okay, so the plants produced by tissue culture will be resistant to different types of diseases. Third important advantage is that many plants can be developed from a single plant. So you can imagine that if you take a single plant, you can get so many meristematic cells from that single plant. So each of this meristematic cell will be forming a group of cell called callus, which is growing into a group of plants. So from one plant, several plants can be produced within a short period of time. And the last advantage is that this technique is commonly used for the propagation of ornamental plants. Ornamental plants means the plants which are used for beautification purpose, which are grown for their beauty, like flowering plants, etc. Okay, so these are the 4 important advantages of tissue culture. First thing, it is a very fast growing technique. Second one, by using this technique, disease resistant plants can be produced. Third advantage is many plants can be developed from a single plant. And the last advantage is this technique is commonly used for the propagation of ornamental plants. So with that, we have come to the end of the asexual means of reproduction. So far we have come to study 7 types of asexual reproduction methods.